Hello and welcome to part 2 of our lecture on the cell. In this portion we're going to be taking a look at organelles and we'll begin with the nucleus. The nucleus is where we find the DNA, that is our genetic material, and essentially rather than having the genetic material within the cell and kind of floating around anywhere, the nucleus wraps a membrane around that genetic material, keeping it separate from the other organelles. There will be a reason why this is important, and we'll talk about that when we address the replication of DNA and the making of proteins later in the semester. But for now, let's just get this idea that it's a storage place for the genetic material. Now, we have a couple of terms we use to describe that genetic material. You can see that in our nucleus up here, which is this big ball with yellow dots on the outside of it, there's something inside called chromatin. And that's DNA, which is wrapped around proteins, but is indistinct. That is, we can't see the 46 chromosomes or 23 pairs of chromosomes which humans have when it's in this form. Chromosomes are condensed genetic material that are being prepared for cell division. So chromosomes are something we will see during the cell division process. We will talk about how humans have 46 chromosomes, better understood as 23 pairs of chromosomes, and that'll have some ramifications both for cellular replication, but also just understanding how we can have two different cell division processes, one that leads to gametes to allow for us to have offspring, and one which allows us to replicate our cells during the rest of our life. And finally inside we have something called a nucleolus, and they're pointing to a single nucleolus here. Sometimes we might use the term nucleoli because there may be more than one, and that's going to be a dark spot. This is a place where we form ribosomal RNA and ribosomal subunits. So ribosomes are another organelle. They're in fact those little yellow dots we see on the outside of the nucleus and associated with this what we call endoplasmic reticulum here. Those little dots are going to be the site of protein formation, but yet those little dots themselves are formed inside the nucleus and their parts are shipped outwards so that they can do their job on the area outside of the nucleus. So ribosomes are our next organelle, and they're different than a lot of our other organelles. Many of our organelles, such as the endoplasmic reticulum, are membranous in nature or surrounded by membranes like the mitochondrion. But ribosomes are not, so we refer to them as being granular, that is, that they're not surrounded by membranes. And ribosomes are most importantly described as the site of protein synthesis, so we will spend some time talking about them later in the semester. They are sometimes associated with ER, or endoplasmic reticulum, and we have two forms of endoplasmic reticulum. Continuous with the outside of the nuclear envelope would be our rough endoplasmic reticulum. And this rough ER is studded with ribosomes, whereas our smooth ER has no ribosomes on it. Again, since both of these are membranes, and membranes are composed of phospholipids, we have the capacity in either of these cases to make something. They're both involved in synthesis. In the case of rough ER, it's protein synthesis. In the case of the smooth ER, it's phospholipid synthesis itself. We can make little vacuoles or bubbles of phospholipids surrounding, in the case of the rough ER, whatever the protein products that are being produced in there are. And that allows us to take these products and ship them somewhere else. Now there are some other functions there. Now since we are making proteins with the rough ER and that they tend to be proteins that are for export, they could be things like enzymes or they could be antibodies in the case of a white blood cell. But in the case of the smooth ER, it has functions like producing estrogen and testosterone. These are hormones that are based off of a cholesterol-like kind of model. So they are more of a lipid than a protein kind of hormone. And it also takes part in something very different, detox. So in the liver, we can use smooth endoplasmic reticulum, for instance, to break down alcohol within our body. This is kind of important in one way in that a way we can respond to alcohol consumption physiologically is to generate more smooth ER in our liver cells so that we can have a more rapid response to detoxify that alcohol. And that's why some people can drink more than others. You can actually build up somewhat of a tolerance. That doesn't mean you won't damage your liver in the long run. It just means that we can explain some of the things we see as far as differences in alcohol tolerance that are independent of size or sex of the individuals involved. Now, associated with the endoplasmic reticulum is the Golgi apparatus, or sometimes called the Golgi complex. And so we can kind of get a nice picture here of what's going on as far as the relationship between these components. Here's our nucleus towards what might be the center of our cell. We have endoplasmic reticulum very close to the nucleus, the rough in particular and some smooth over in another area. And these can produce lipids or they can produce proteins, and they ship them off to this Golgi apparatus. So we think of this as a site of modification for export of these proteins and lipids. And you can see here I mentioned it forms glycoproteins and glycolipids. So the glyco component suggests that we are adding various sugars to either one of these. And we will have both glycoproteins and glycolipids, which may be part of our plasma membrane on the outside of the cell, so they must be exported there, or which can be shipped off or secreted. So you can see here that the Golgi apparatus can sort and package these in vesicles, such as this secretory vesicle, and then they can actually secrete some of the products outside of the cell to be distributed throughout the body. Over on the other side, we see something else interesting that happens, and it relates to our next term here, the lysosome. So a lysosome is produced by the Golgi apparatus, and it contains digestive enzymes. Now, often we will specifically say that these are hydrolytic enzymes, and we know that hydrolysis is the process by which we take a big molecule and break it down into smaller components. And they give you a good example here. We have what looks to be a 4-carbon molecule. It's unclear exactly what's going on, but we bring that in an incoming vesicle. We fuse our lysosome to it. Those membranes open up so that our enzymes can mix with this molecule and break it down into its subcomponents. And then, of course, we can dissolve that vesicle. We can release those subcomponents into the cell, and we can utilize them for cellular processes. Now, this will all be involved, say, if we're trying to get energy out of sugars. We can bring sugars in, and our first step is to break these things down. Usually, we're just going to talk about a simple process of breaking them in half and generating a couple of ATP. Now, obviously, we're going to talk later about how if we have mitochondria, we can take additional steps and generate more ATP. Before we talk about generating energy, though, we're going to talk about some movement and a little bit about structure. So there is a structural component to cells that does a little bit more than, say, our skeleton does, but we call it the cytoskeleton. It's protein fibers, and if we think about our skeleton is really about providing shape and then providing some levers for movement. Now, of course, we have muscles that attach to those levers in our skeleton to allow movement to occur, but these protein fibers themselves are also a little bit like muscles. That is, we have some of them that are capable of changing their size. So the cytoskeleton is not just about providing shape or even anchoring us to adjacent cells, but it's also about moving things around, and that could be moving around organelles or moving around chromosomes during cell division. Now, we do also have two very specific organelles that are associated with movement, cilia and flagella. Or we could use the singular terms, cilium and flagellum. Interestingly enough, we need the single term for flagella, that is the term flagellum, because something like a sperm has a single flagellum, and that's the only place we have flagella in our body. Now, cilia tend to occur in multiples, and you can see kind of a carpet down here, looks sort of like an anemone. And cilia, since they occur in multiples, means that we rarely are going to use the singular form of that word. What's interesting about this is what they have in common as well as what they have different. So this flagellum has been sliced into to allow us to see microtubules arranged around this. And they have a very specific 9-to-2 pattern where there are bundles of pairs on the outside and a pair on the inside. And this particular structure here is going to be kind of a motor. It's going to be able to flip back and forth. The cilia can do the same kind of thing, but they are much shorter and again occur in multiples. We're going to find cilia on the outsides of some of our epithelia. That is, these tissues that are going to line our body, so that we have linings like in the case of our respiratory system, where we can use cilia to move mucus. which has captured some of the inhaled particles that we don't want to have move all the way down into our lungs. Now, we can't really talk about the cytoskeleton without also addressing something called the extracellular matrix, which is kind of like an extension of that on the outside of the cell. And this is going to help to bind some cells together. So we'll talk kind of at some point about what we call junctions, where cells will connect to each other, and they do so in different ways depending on what the needs of that tissue are. But we can have this connection between the inside and the outside, and the extracellular matrix will be these proteins and polysaccharides, that is big sugar kind of chains, which are going to help to create binding to other cells, or in the case of a connective tissue, since the extracellular matrix is going to be so abundant, is actually going to create the characteristic behavior and structure of that particular tissue. That is, a connective tissue is not like our other kinds of tissues. Instead of being cells bound together by a small amount of extracellular matrix, it's a large amount of extracellular matrix produced by a smaller amount of cells. All right, and here we see some junctions. So junctions, again, are components of this which are going to bind cells to each other, and the needs of each tissue are a little bit different, so we have different kinds of junctions. A common one is what we call an adhesion junction, or sometimes we'll use the term desmosome in here. These are going to bind cells together. Basically, they're going to have filaments of cytoskeleton reaching across, so we call those intercellular filaments. We've got filaments of cytoskeleton on the inside, and we are basically loosely binding some cells together, and they're going to allow some degree of movement, and flow can occur between adjacent cells because they are not so tightly bound together. Obviously, a tight junction is a good name to describe the fact that some cells are bound more tightly together. And you can see that these have proteins which bind the plasma membranes of adjacent cells together, and that's going to prevent flow between the cells. So if we prevent flow, that's going to create more of a waterproof type barrier, and we tend to find this in areas like the distal convoluted tubule of our urinary system as well as our bladder, keeping that urine in place and not allowing it to seep out into surrounding tissues. Gap junctions are particularly interesting, and we see those over here. We have membrane channels in our protein, and they connect from one cell to another. So rather than being about flow between cells, it's flow from one cell to another. That is, we can actually have an ion move from one cell into the next cell over, essentially passing along electrical or chemical signals. And we're going to see gap junctions in things like our heart muscle, which ensures that our heart is going to contract in unison so that we can efficiently eject blood from it. Okay, so we'll get a little bit about metabolism in here, and this is going to relate to our use of sugar to produce ATP. But first of all, metabolism is just all of our chemical reactions in our body. And so obviously what's going to happen in our body is we're going to bring in nutrients, we're going to break those down and make energy available, but then we're going to utilize that energy to build up things as well. Say if we need to make some proteins, or if we need to make some lipids, if we need to make whole new cells, all that's going to require energy. And we get what we call a metabolic pathway in our body, and we can see that down below here. So in this case, the letters are reactants or products. They're actually both in most cases. We start out with a substance A, and you can see here that that's going to be a what we call reactant, or maybe we might call it in an enzymatic reaction a substrate. That's a particular molecule which is going to become something else, which we call the products. But in this case, A becomes B. But then B is going to undergo a reaction to become C. Now that's not quite the end of this metabolic pathway. We have to understand what these enzymes are as well. And enzymes are catalysts. They just happen to be biological catalysts, which are generally made of protein, but we have forms of RNA that also act as enzymes. So in this case, these enzymes speed up reactions. They make reactions happen more quickly. They make reactions happen with less energy than they would otherwise require to get started. They may occur at lower temperatures then. And in this case, you can see that the substrate enters the enzyme, forms what we call an enzyme-substrate complex. We form our products. In this case, it looks like we're undergoing hydrolysis. So what we're doing is we are breaking this substance down into smaller substances, and then we release those products. What's so interesting about the enzyme is that the enzyme is released unharmed and can do this process again. So in our metabolic pathway down here, A is converted to B, but only in the presence of enzyme number 1 here. B is converted to C, but only in the presence of enzyme number 2. So in order to get from A to G, we have many metabolic pathways which may have a lot of steps. And as you can imagine, if you need product G to live, there are a lot of things you might need. You're going to need, first of all, A, which is ultimately going to be the substance that allows you to initially work your way through this pathway. And if your diet is deficient in A and you can't produce it on your own, you wouldn't be able to get to G. But we also have to have enzymes 1, 2, 3, 4, 5, and 6. And if you have genetic defects which cause you to be unable to produce those enzymes, you would not be able to get through this metabolic pathway. So we can get a little bit of an idea about how genetic disorders may arise. Sometimes they do interfere with metabolic pathways like this. Sometimes they actually interfere with structural components of, say, proteins that are in a cell and are essential for function. So they can do more than one thing, but it's pretty easy for a number of different defects to cause the same kind of problem. That's what a metabolic pathway really tells us. All right, so how do these enzymes work? Well, they have an active site. The active site is the place that those substrates will bind to. In this case, we're looking at synthesis, and that's going to be something we're going to do through dehydration. We've got substrates binding into the active site, and the active site has what we call specificity. That is, it fits particular substrates, and it doesn't fit other things. It's going to convert these into the enzyme-substrate complex, eventually release the product. This is a single molecule now, and the enzyme can do this all over again. But what that means is that generally an enzyme can work for a particular reaction, although it may occasionally work for a very closely related substance. And this can be kind of important because we may have something we call inhibition, and there's such a thing called competitive inhibition, where if we have two substances, we can have competition for the enzymes, and that will slow down any one of those particular reactions. All right, so what again is it that enzymes do? Well, they lower the amount of energy needed, and we call that the activation energy. And so what we're going to get an idea of here is that there's a certain amount of energy available. We have an energy of our reactant, this thing that's going to go into the reaction. We've called it the substrate in the last couple of pictures. And then we have the amount of energy in a product. And the difference between those is energy that can potentially be released. But what you can see in here is that we need to have a certain amount of energy called the energy of activation that needs to be added in order for the reaction to take place at all. So even though our product will hold less energy, meaning that there should be energy released, we need to put energy in to get the reaction to take place. And the key thing about enzymes is that they will lower this energy of activation. And you can see that here, that we have a much smaller hump to get over. What that means is that we will have a greater yield of energy. Okay? And also, in other circumstances, we may not even have the reaction take place with that large energy of activation. So, activation energy, important. It's the energy that must be added to cause a reaction. It takes place whether we have to put energy in to make a higher energy product, or if we gain energy by breaking a high energy reactant down into a lower energy product. And enzymes will often need assistance. We call the things that do that coenzymes. These are molecules that assist enzymes, and one of the prominent ways in which they do this is by accepting or donating atoms or electrons. And so we're going to take a look at NAD+ in a moment as being a coenzyme which assists us with our glycolysis process. All right, so what do we want to know about energy? Well, we want to address something called cellular respiration, and I like to think of this as the combination of glycolysis, a reaction we'll talk about, and cellular respiration, which basically continues what glycolysis starts under some circumstances. And so what this really is this process here of taking a sugar, glucose, adding oxygen, in this case it's 6 oxygen molecules, through multiple steps, which is why we have multiple arrows. We're going to convert this into 6 carbon dioxide and 6 water, as well as ATP, and following what we call the second law of thermodynamics, we're going to produce heat. So the second law of thermodynamics, which will become important in ecology later on, basically states that when we convert energy from one form, that would be glucose, to another, the energy in the bonds of ATP, we're never completely efficient in doing this, and some of that energy will be released in a less useful form, and that's going to be heat, which is basically a measure of molecular movement and can be used, interestingly enough, to increase molecular movement, making us have a warmer body temperature, which can make reactions occur more easily. So it's not entirely useless. Now I want to contrast this glycolysis and cellular respiration with photosynthesis. And we see a nice diagram here which really tells us how life on Earth largely works. Sunlight energy comes in, chloroplasts can undergo photosynthesis, and what they can do is take simple molecules like carbon dioxide and water and form organic molecules like glucose, and oxygen is produced as a byproduct. It ends up that glucose and oxygen is required for cellular respiration so that we can generate ATP, our form of energy, and in the process we form CO2 and water, which are the same things that plants are going to need along with that sunlight energy to be able to produce more of that glucose and oxygen. So it's a nice circle there, and these things work great together. The reactions are definitely different if we look at them on a very small scale, but when you look at them in a big scale from the outside, they are basically the reverse of each other. That is, if we wanted to figure out how photosynthesis takes place, we could largely just turn the arrows in our bottom equation around to face the other direction. Alright, so how do we generate energy? We're going to start with glycolysis, and that is the process of taking a glucose, which we're going to bring into our cell, and we're going to break that down into 2-pyruvate. In order to do this, we're going to need NAD+, this is our coenzyme, and we're going to convert that into NADH, which has picked up a hydrogen and 2 electrons. In this process, we can turn 2-ADP, adenosine diphosphate, into 2-ATP, which we can use for any cellular processes. So what's good about this? Well, it's anaerobic. It does not require oxygen, meaning that we can do this even if we lack oxygen, even if we lack mitochondria. So we think of this as an ancient metabolic pathway. In other words, this is kind of the first step in taking molecules like glucose and generating energy from them. Why do we think this? Well, glycolysis occurs universally. So it's associated with a process called fermentation that we'll need to learn about, and with cellular respiration. It doesn't require oxygen, and of course there wasn't free oxygen really available on the early Earth for us to be able to do this kind of thing. And it does not occur in a membrane-bound organelle, the mitochondrion. So that means that even if you lack mitochondria, if you're a little bit more simple, you can still do the process of glycolysis to generate ATP. Alright, so if no oxygen is present, glycolysis will be followed by fermentation. And fermentation is essentially this, at least in us. We're going to take 2-pyruvate, a 3-carbon molecule. You can see the glucose, of course, is a 6-carbon molecule. And we're actually going to convert it into another couple of 3-carbon molecules called lactate. You may be familiar with lactic acid and the association people have with that and sore muscles. In this case, what's happening, we're taking our NADH and we are converting it back into NAD+. So we need to do something with that hydrogen and those 2 electrons so that we can get our NAD+ back so we can continue to do glycolysis and to generate ATP. That is that the fermentation, this second component of this, isn't actually generating any ATP itself, but it allows us to continue the glycolysis process to generate 2-ATP. It's anaerobic. Lactate can be toxic to cells. It will build up. There become a problem with that. Not everything does this particular pathway though. Bacteria and yeast form alcohol and carbon dioxide instead when they do fermentation. So when we make beer or wine or bread, any of those kind of things, we have fermentation occurring and we're taking advantage of either producing alcohol or, in the case of rising bread dough, the production of carbon dioxide. But they're doing it for the same purpose, to regenerate this coenzyme to continue glycolysis, to continue forming ATP. So again, when do we ferment? When we don't have oxygen, or if we're an organism that doesn't have access to oxygen or doesn't have mitochondria. But if we have mitochondria, the organelle that is responsible for synthesizing ATP in the presence of oxygen, and we have oxygen, we can undergo cellular respiration. And cellular respiration is quite a complex process. We're going to kind of make it a little bit more simple, but cellular respiration is just a series of reactions that are going to break glucose down, and we can generate overall, rather than 2 ATP, somewhere around a total of 32 ATP. Now, we used to think that we could make 36 ATP, or in a couple of cell types 38 ATP, but we've scaled these back in recent years. So, what do we do for this process? The complete process by which we're going to use oxygen and mitochondria to generate ATP is going to start with glycolysis. We form NADH and we form ATP. We're then going to go into a phase called the citric acid or Krebs cycle, where we form more NADH and something related called FADH2. As you can imagine, FADH2 is also something that's going to carry hydrogens and electrons. And we also start producing the CO2, which is going to be our waste product from this process. And then we get into the electron transport chain. In this case, what we're going to do is convert the energy from those coenzymes, from the electrons they are carrying, into ATP. And that's really where the vast majority of this energy comes from. So up to this point, we haven't produced a lot of ATP, but with the presence of oxygen, we can produce water and use the electrons carried by NADH and FADH2 to generate loads of ATP, making cellular respiration essentially much more productive than fermentation. Okay, so the idea here is this. If we take a look at this picture down here, we take energy from cellular respiration, we produce ATP, we use ATP, and the energy from ATP breakdown is used for metabolic work, muscle contractions, secretions, active transport, and then that leaves us with ADP, adenosine diphosphate, and free phosphates, which we must recharge using energy again from cellular respiration. And we could get a little bit of an idea of how this all works by taking a look at this picture here. We've got a blood capillary outside of our cells, and glucose is going to exit those. Of course, we're going to have some cellular junctions here, which are going to allow for the flow of glucose between them. We may have a lot of molecules just diffuse directly across the thin plasma membranes of this capillary, and then these things are going to enter the cell. Obviously, oxygen can do so through diffusion directly across the phospholipid bilayer, but glucose is going to have to go through using facilitated transport and using a protein carrier. Now, the thing about this is we keep bringing glucose here, so we're going to have high concentrations outside, and we use it on the inside, so we're going to continuously have low concentrations. So we undergo glycolysis, and glycolysis, as we remember, takes place in the cytoplasm. We do not need a mitochondrion for that, and it does not take place in the mitochondrion. But if we have a mitochondrion and we have oxygen, we can do better than this 2 ATP that we generate from glycolysis. Instead of going through fermentation to regenerate our NAD+, we take our NADH produced in glycolysis and send that to the mitochondrion. We also send our pyruvate, that half of a glucose, into our citric acid cycle here, and we can run that through. There's actually another preceding step that some textbooks would talk about. Basically, the idea here is that through a previous step and through the citric acid cycle, we're going to take the 3-carbon pyruvate and break that down into carbon dioxide, which has a single carbon. And so for each pyruvate, we should get 3 of those CO2, and in that process produce either a little bit of ATP or some NADH, and of course some FADH2. Those electron carriers or coenzymes are transported to what we call the electron transport chain within the mitochondrion, and that's where the majority of our ATP is generated. Again, that's also the point at which oxygen is required and the point at which we're going to be forming water by taking hydrogen from the NADH and oxygen to form the waters. All right, and here's another diagram that just shows us that same kind of process with a slight bit more detail. So this should be the last time we have to look at it, but it'll show us, I think, a nice complete view of the breakdown processes, perhaps in more detail than we want right now, but it's good to know what kind of detail people understand this in. And of course, we understand proteins involved in many of these different steps, so the detail is much greater than what we see here. But we begin with glycolysis, break glucose into 2-pyruvate, get a little bit of ATP, get some NADH. We go through what we call pyruvate oxidation, which is what was missing on the last diagram. We produce 2-acetyl-CoA, which are going to be a 2-carbon molecule, and we produce CO2. We also produce some more NADH, and I like to think of NADH and FADH2 as future ATP. It's not ATP yet, but we will make it into ATP. In the citric acid cycle, we take those acetyl-CoA, break them in half to make CO2, and we generate a little bit of ATP, but we generate a lot of future ATP. And through what we call oxidative phosphorylation, the processes that take place with electron transport, those are going to use our oxygen, produce water, and produce a lot of ATP, giving us the vast majority of our ATP in this very final step where the oxygen is actually required. All right, so one more kind of idea to relate eating to this idea of generating ATP. We don't always just eat glucose. Okay, so that's one of the important things here. And we ask a simple question here. Can we gain energy from lipids and proteins? The answer is yes. And the answer to how is that we use the same mechanism. We use things like glycolysis. We use the citric acid cycle. So we can use those same things to break down different kinds of molecules. And here's a good example. Something like a pizza will have proteins, carbohydrates, and fats. The carbohydrates are broken down into glucose and do just what we've talked about, glycolysis, going to making some acetyl-CoA, going to the citric acid cycle, and electron transport, and we get a lot of ATP. With proteins, your amino acids can jump in at multiple different steps here depending on which pieces we break them down into, and we can go through the electron transport chain and generate ATP. With fats, it depends whether we're talking about the glycerol of our triglyceride or the 3 fatty acid tails. We can either drop the glycerol into the glycolysis process or the fatty acid tails later on here to where we break them down into acetyl-CoA, go through the citric acid cycle, and the electron transport chain. What this doesn't address is something that's a little bit even more crazy. Not only can we get energy from all different food sources through different ways in which we use this same mechanism, but you can actually use this mechanism backwards in parts to generate some of these molecules.